All right, everyone. I am here as just hanging out, making sure everyone has a good time today. And I only see my face. I don't know why. I want to view everyone. There we go, gallery. Okay. So guys, we're here today. I'm so happy you could join. Thank you for those of you who don't know what Blood Pack is or just got this message and were told to hang out with us today by your colleagues. Welcome. We are a consortium of about a little over 80 organizations, pharmaceutical, to talk about all the things that we may have missed. So we are laser focused on the deliverables that we have for our existing working groups, but over the course of the year, science moves forward, things develop, and what should we be paying attention to as we move forward into the next year? And so we are very lucky to have an extraordinary group of scientific co-chairs who pull together this discussion, have a suggestion for what the topic should be. They focus on that at the very beginning of the year. And then we are very lucky to have a few handful of volunteers who really help mold this conversation throughout the course of the year as well, and kind of take it to today. I have to give a special, tremendous thank you to Doris, who works for Blood Pack, who actually coordinates this entire conversation. Please, everyone, people do not need to look so serious. This should be a dialogue, a conversation. You're going to hear a few different speakers, and then you have the opportunity to listen to a panel conversation and put your questions in the chat. I'm going to keep everyone muted. You do have the power to unmute yourself, but try not to unless you have something to say important. I mean, I rarely do, but you might. Those are the rules of the game, and I will come back on at the very last moment to make sure we're done here by 3 o'clock. Does that work for everyone? Yeah, Robert gets first positive reinforcement, 10 points and a gold star. Asaf, I am handing it over to you, my friend.
Hello, everyone. I hope you see my screen. Thank you for joining us for this exciting event. It's my pleasure to kick off this meeting about therapy selection. My name is Asaf Zviran. I'm the co-founder and CEO of Prism AI Therapeutics. Previously, I co-founded and led C2I Genomics, which introduced one of the leading paradigms for residual disease monitoring using whole genome sequencing. At Prism AI, our focus is on addressing fundamental challenges in multimodal clinical machine learning, particularly around the derivation of mechanistic molecular models, biomarkers, and companion diagnostic for multidimensional clinical trial data across various indications: oncology, mental health, inflammatory, and neurodegenerative. Despite the central role of companion diagnostic in oncology, more than half of the patients still lack a therapy-associated biomarker. Over 30% of those treated with CDX-matched therapies fail to respond, and large majority of drug development programs are terminated due to lack of efficacy. We believe that the integration of genomic, transcriptomic, epigenetic, digital pathology, and functional assays present an unprecedented opportunity to capture the full biological complexity of disease-host-drug interaction and identify robust models and biomarkers that expand the accessibility to therapy selection to all patients. The critical question that we'll discuss today is how close are we to realizing this vision? The following discussion will explore emerging frontiers in multi-omics and AI-driven therapy selection across tissue and liquid biopsy modalities that are moving the field toward this goal. We have a really great lineup of industry leaders speaking today. The first one, Tuvik Becker, CEO of Pangea Biomed, is going to talk about InLight, a unified response prediction platform. Tuvik, you can start.
Thank you, Asaf. Let me share the deck. Okay, I hope you can see my screen. So good afternoon, everyone, and thank you, Asaf, Lauren, and the whole Blood Pack team. It's a real pleasure and privilege to be part of this wonderful day. My name is Tuvik Becker. I'm the CEO of Pangea Biomed, where we try to solve one of the major issues in cancer treatment. We've built InLight, a biologically informed AI platform that unifies data from sequencing, pathology, and hopefully soon also liquid biopsies, to make therapy selection more personalized, more accessible, and more adaptive. Genomic profiling transformed oncology, as we all know, and novel targeted drugs and immune checkpoint blockers can be very effective. In some cases, these drugs offer curative treatment to metastatic disease, which is something that we could only dream of 30 years ago. But if we look at the bottom line numbers, only 30 to 40% of the patients have actionable mutations discovered through NGS, and few of them actually benefit directly from matched therapy. Furthermore, existing biomarkers are siloed by modalities, and they show a low level of overlap. For the most part, they don't seem to capture the underlying biology of the disease. So we really need an integrative layer that can connect the genomic layer, the transcriptomic input, and morphological data into a more unified prediction engine, and that's what we're trying to do with Enlight. Enlight has been developed over the last 6 years in very close collaboration with the National Cancer Institute, and it analyzes gene interaction networks to quantify how the biology of a specific tumor aligns with the mechanism of action of various drugs. The underlying intuition is based on synthetic lethality and synthetic rescue principles rather than on purely correlative models. So with that, Enlight builds predictive biomarkers that take into account not just the drug's direct targets, but the broader tumor molecular environment that would determine response or resistance to a particular treatment. It provides the Enlight matching score, which is a standardized response score that spans different cancer types, different drugs, and different input modalities, and also different treatment modalities. Importantly, the biology-informed approach of Enlight doesn't require training on large cohorts with matched pre-treatment data and post-treatment outcomes. So, of course, we train on very large cancer data, but this is more generic data that can be applied later to any drug that we want to predict response to. And that way, we relieve one of the main hurdles for the development of AI-driven response prediction models. Enlight NGS, which is our first generation technology, uses RNA sequencing as the input and has been already validated on thousands of patients with diverse cancer types and treatments. In this chart, we see a subset of around 1,000 cases in 30 different clinical trial data sets spanning 10 different cancer types and 16 different checkpoint blockers, TKIs, and monoclonal antibodies. In this aggregated cohort, the overall odds ratio of response between Enlight matched and Enlight unmatched treatments is 2.64. In other words, patients who receive Enlight matched treatments have much better chances of response than those who receive treatments that were Enlight unmatched. Enlight NGS is now used clinically for patients who have exhausted the standard treatment options, expanding access to precision medicine beyond mutation-driven matching. But the evidence we have at this point is not limited to retrospective data. Enlight matched treatments have already saved the lives of patients by offering recommendations that contradict, in many cases, standard biomarkers and guidelines. Here's the story of one patient who was one of the first among these cases. And here I need to disconnect and reconnect again in order to enable audio. And in the meantime, I'll just say that the audio for this is in Hebrew, but there are captions. So please read the captions unless you speak Hebrew. Okay, let's see if this works.
למעשה אמרו לי בבית חולים האחר שאני פשוט צריכה לחכות למות.
אני מציע לשלוח את הבדיקה של ה-RNA לחברה סטארט-אפ ישראלית, קוראים לה פנגאה. אחרי מספר ימים הם חוזרים עם תשובה שייתכן וטיפול אימונותרפי עם 2 תרופות יכול לסייע לטל.
הטיפול עבד והגרורות פחתו, ועכשיו אני במצב שאין בכלל זכר למחלה שהייתה. אומרים לך שאין לך סיכוי לחיות ואז יש חיים, אז הרגשה מדהימה.
Yeah, so by the way, Tal is now, she celebrated her 40th birthday not very long ago. It's been 5.5 years now since this case was first brought to us. Anyway, the newer line of tests that we've developed is called EnlightDP, standing for digital pathology, and it combines Enlight's biological engine with deep learning based digital pathology. So instead of taking a tumor block, running it through a sequencing machine to generate read counts, what we do is we trained deep learning architecture called DPT to translate the morphological features in standard H&E slide scans into inferred gene expression profiles that then feed into the same Enlight brain, so to speak. So with that, EnlightDP makes treatment prediction possible from archival tissue samples, enabling analysis even when sequencing isn't feasible with very, very fast processing times. EnlightDP was introduced last year in Nature Cancer in July of 2024 as a general framework, and since then we published several additional validations, 2 peer-reviewed journals, articles, and 5 major conference presentations, another one coming up this weekend at CTC. We've already shown that the same underlying algorithm works across 5 different cancer types and in very different clinical scenarios in retrospective as well as prospective setups. One of these prospective setups is a currently ongoing trial at Sheba Medical Center, where EnlightDP is being used to guide the choice of therapy for testing using a 3D ex vivo functional assay. A really cool assay, by the way, developed by our colleague Ronit Sachi-Fainaro from Tel Aviv University. In the trial, slides from resections or biopsies are analyzed by EnlightDP within 24 hours of collection, and top-matched drugs are added to the functional assay alongside the drugs from the list provided by the physician, what they thought should be tested using this ex vivo system. And the surprising result is that in more than 70% of the cases, EnlightDP uniquely suggested targeted therapies that proved sensitive in the 3D models. Enlight was able to correctly predict response in 2/3 of the cases where outcome data has already become available, and there were already 2 cases where non-standard Enlight matched therapy was prescribed simply in the absence of any conventional alternative. Nothing else came up as effective in the 3D assay, and in both cases this resulted in response. So in one of these cases, the patient had 3 previous treatments on which she progressed, and then with the non-standard treatment that Enlight brought up, she achieved more than 10 months of remission. Our next step is to move from compassionate and exploratory use to a structured testing pipeline across major indications. Each of the rows in this table represents a clinical question that we're currently addressing specifically, from predicting non-responders to immunotherapy in head and neck cancer, responders and non-responders, choosing between IO-IO combo and IO-TKI in metastatic RCC, or identifying exceptional responders to hormonal therapy in HER2-positive breast cancer. Altogether, these studies represent more than 600,000 potential patients per year in the US and the European Union who could benefit from specific ENLIGHT-based decision support tests. ENLIGHT-EP is no longer just a discovery platform, it's transitioning gradually into a clinical decision tool, indication by indication. Having demonstrated that ENLIGHT works across sequencing and pathology, the natural next step for us would be extending this to liquid biopsy with a solution that we call ENLIGHT-LB. The same gene interaction network can theoretically interpret cell-free RNA patterns in peripheral blood or other readouts that can teach us about gene activity in the tumor, linking these back to drug mechanisms. And we've been working on developing this technology as part of the Liquid BX consortium, which is funded by the Israel Innovation Authority. Such a solution would really allow therapy matching from a simple blood draw and make precision oncology both non-invasive and repeatable. So why go multimodal? In my opinion, tumor heterogeneity, both in space and in time, is one of our biggest adversaries at the metastatic stage of cancer. A single biopsy captures one region at one moment, but as we all know, tumors evolve and they diversify under treatment pressures and in general. So by combining modalities, histopathology for the spatial context, RNA sequencing for the molecular basis, and liquid biopsies for temporal dynamics, ENLIGHT will be able to capture the tumor's biology far more completely. That's our vision for how multimodal precision oncology could become not just descriptive, but truly adaptive. ENLIGHT NGS, our first solution, provided the scientific foundation, the biology-driven algorithm for individually matching patients with treatments. With ENLIGHT-DP, we brought that approach into the world of digital pathology, enabling much more practical deployment in any hospital with a good slide scanner. ENLIGHT-LB will hopefully extend it even further, turning precision oncology into a continuous process, from initial tissue analysis to serial liquid monitoring. So one algorithm, three modalities, driving a single goal: to make functional precision oncology accessible to every patient. Before I wrap up, I would like to mention the wonderful group of renowned scientific advisors who are helping us develop, refine, validate ENLIGHT. It's really a great privilege to be working with such a team of clinicians and cancer researchers. Finally, thanks to the amazing team at Pangea who's made all this possible, and to the many others who've collaborated with us on the works featured in this presentation. To summarize, ENLIGHT is evolving into a unified predictive ecosystem. It bridges the gap between genomic, morphological, and now also blood-based data, delivering one coherent view of how each patient's tumor is likely to respond to treatment. We are really looking for collaborators to help us develop ENLIGHT-LB in particular, integrate multimodal data streams, and bring this into the clinic. Thank you very much, and I'd be very happy to take questions later.
Amazing. Thank you, Tuvik. Really congratulations, especially on the impact on patient lives. Really amazing. Regarding questions, so just to remind everyone, you can put your question in the chat, and there is going to be a Q&A session afterwards. So please put your question. We are going to come back to it later. Next speaker, Alexander Savenchko, head of diagnostic and molecular pathology in Novartis, going to speak about empowering innovation with digital pathology solutions. Alexander.
Thank you. I hope you can see my screen. Just want to make sure.
Yes.
Right? Okay, good. Yeah, good afternoon, everyone. Thank you. Yeah, my name is Alexander. I'm actually representing Novartis today. and I'm honored to present today as a few data that actually we're going to show the empowering of the clinical innovation with digital pathology solutions. So before I, sorry, just a second. Okay, good. Before my presentation, just a few disclaimers, so that please know that this presentation is based on publicly available data only. And this data is actually was generated for educational purpose. And I also would like to kind of highlight that the information is kind of accurate and the best of my knowledge, but does not necessarily represent Novartis' official positions. So for today, I would like to touch base on a few, I would say, like critical subjects. So first, I would like to discuss the benefits of digital pathology and the deployment in routine practice. I also would like to present 3 cases based on the work that we did here. And the first case we're going to actually represents the generation of the tumor mask that we are able to generate with the reference endpoints. As a second case, I would like to show some examples of the immunophenotyping and what digital pathology can do with additional features like cell-to-cell interactions or neighborhood analysis. And as the last case, I would like to briefly present one of our recent work on the PD-L1-IHC. It's one of the most well-known CDX and using like digital image analysis to enhance the diagnostical capabilities. And we specifically kind of collaborate with a few investigators on the gastric cancer patients.
Alexander,
So
sorry
with
to
this,
disrupt, but we don't see the slides. Maybe you want to try to share them again,
oh,
or
I'm
it's
sorry.
maybe just me.
I can see them.
I see the slides.
I can see the
I
slides.
can see them.
Okay, so it's just the problem is okay.
Oh, okay. So just want to make sure. Okay, because yeah, I can see them too. There, it's currently shared. Okay, can I continue? Right. So is it okay?
Yes.
Okay,
Yes,
good. So I think there are a lot of discussion around like what digital pathology can do, and with this slide and actually another slide, so like 2 slides, I'm trying to summarize what digital pathology can offer. So first of all, there are a couple advantages, right? And as the pathologist by training, I'm always going to say that, yes, I have some limited scope of assessment, especially when it comes to quantitative evaluation. So with digital pathology solutions, so it's actually clear that we can analyze the multi-parametric biomarker assessment. As I mentioned, so what I'm really excited about some additional parameters like cell-to-cell interactions, which is easy to deliver with the specific algorithms. It has like a really high throughput and cost efficient in terms of the turnaround time, which is quite critical, especially in the clinical science. So the procedure, once the algorithm is fully deployed, validated, it can be automated, right? So that means that we only have like, the supports from histotech and pathologists to do either QC process or overseeing the quantitative workflow, which is again like very efficient in terms of the clinical operations. And what most important, it actually has like a high reproducibility and accuracy, which is again critical in the late stage clinical development trials. And also it can be like securely store the data, allows more like collaborative efforts between like local sites, central lab, and facilitates collaboration with academic centers. There a lot of work has been done in translational science, right? And sometimes when we're hearing about some of the exciting data on translational science, it's not necessary that this is going to be applicable in the clinical science. So again, here I'm trying to kind of summarize the benefits on the translational clinical science. But please keep in mind that some of the clinical operations could be a critical component. So as I mentioned, so the telepathology is one of the steps that Novartis is really like working hard to implement. And we do have a few clinical trials, few examples, few collaborators, a few vendors that already has implemented the learning tools, the tools that actually allow us to share results with the local sites in a very quick turnaround time, to collaborate with academic centers in multiple indications. So I'm quite positive about the approach of the telepathology in the clinical science. As I mentioned before, the data throughput is actually critical. And especially like in the phase 3 study, when you have like quite significant amount of patients and data to be analyzed. So the data throughput and turnaround time could be critical. And again, a digital image analysis and digital pathology can successfully address those points. And of course, the automation process, right? So to eliminating any kind of human factors, even like we are all human beings and being a pathologist. So I think it's kind of critical step to make sure that the data will going to be robust and accurate enough. So now I would like to really focus on a few examples, just to kind of convince you, maybe not convince you, but show like you more data that actually can be generated with a few kind of simple steps and few simple tasks. So here is one of the examples where the digital tissue annotation I can hold promise to replace conventional morphological diagnostical staining. Here you can see on the left the pan-CK staining, the pan-cytokeratin is actually going to stain the tumor cells based on the epithelial component. And then we used the halo classifier from the company called Indica Lab, so it's a registered trademark, that actually can detect not only morphological features, but also the staining intensity, positivity, the pattern. And based on this algorithm, actually, without any reference, once we applied it on consecutive slides, so this classifier actually shows quite robust performance. So with this kind of, we are positive that, like sometimes when you have a denominator in the tissue and collecting the tissue from the tissue samples from the patient, it's quite not easy to evaluate invasive margin, to evaluate, the area of interest, and to apply in the classifier or a tumor mask or tissue mask could be a very nice and useful tool that we at Novartis using quite frequently right now. As this example, like this is the study that has been published back in 2021 in the phase 3 study, where we're able to implement this tool with additional cell-to-cell interaction analysis in melanoma patients. So what actually has been done? So as I said, the tumor mask can actually visualize the invasive margin. And here's like on a rainbow scale, you can see like the intervals for the invasive margin and depositions of CD8 or T effector cells, right? So that is quite important in immunotherapy. But it's hard to really predict, like what will be the response from these particular patients unless you know how the T effector cells are going to behave. And this analysis actually shows you like in a cellular base, the interaction between tumor cells and T effector cells. And actually it helps to clinically validate and be like as a prospective PD biomarker in the trial. And I'm happy to say that this actually work has been successfully implemented as part of the phase 3 study under regulatory environment. That means that Novartis were able to develop, deploy, validate this particular digital image algorithm to meet all regulatory standards. Even more, and this is actually one of the exciting parts of this presentation that I'm happy to share, there are a lot of discussion about what kind of tumor we can call immunogenic, right? So what's the immunophenotyping of the tumor? And there are a lot of different criteria, classifications, and there are no kind of clear standards. Here with the tool that I just presented to you, we are able to kind of take the whole entire lesions and to really understand, what is happening in the tumor, right? So how the actually tumor behaved and what's the number of the T effector cells in the tumor lesions, in a stromal lesions, and what's the interactions between those cells. And again, what's the predictions based on the therapy outcome we might have. So here is like a 3 phenotypes. The inflamed, when we actually have like T effector cells present in the stromal compartment, and fully engaged in the tumor, with the tumor being like recruiting by T effector cells. The phenotype called excluded, that means that you don't really see much T effector cells or any cells of interest, it could be, right, in the tumor lesions, but most of the cells are located outside. And the desert, when there are any or no sign of T effector cells or any other cells of interest, recruitment either on the stromal or tumoral compartment can be observed. So it's kind of easy tool. Of course, it's validated not only analytically but clinically with specific thresholds to be applied. And again, this tool was successfully implemented as part of the phase 3 study. So speaking a little bit more on the routine part of the CDX assessment, for example, right? And the PD-L1 is well known, and there are a lot of publications in lung, in melanoma, right? So showing, okay, what's the PD-L1 cutoff we have to use, how to handle this work. There are a lot of publications showing that, okay, the CDX might not be fully available on some of the sites, and there are a lot of lab-developed test, LDT, has been introduced to the field. And gastric cancer is one of the indications that we as a company had an interest to discover on what PD-L1 tool can be more useful in terms of the multiple parameters that we assessed in the clinical practice. So this is just a brief snapshot on what assay has been used. And there are like CDX in different platforms. There are two major scoring guidance that I don't want to go into much into details, but there are like two guidance that pathologists used all the time on the PD-L1, independent on indications. And it's like when you really have like combined positive score, means like enumeration of the positive cells in denomination of tissue of interest. And when you have the area assessment called like tumor area positivity or TAP scoring, and this could be applied for any kind of tumors when you have the percentage of the positive lesions. So what we did at Novartis, and again, so we are trying to be efficient in terms of the work, considering that every single staining, and of course we cannot change the strategy that local sites or hospitals will run because they have the strategy based on the preference, on the availability of the markers, on the multiple other factors. And here we can have like consecutive sections stained like, side by side. And you can see the difference, without even being pathologists, you can say, okay, this is more like dark, this is more like a lighter, this somewhere in between. So how we can harmonize, the staining and how we can be ensure that all of these PD-L1 stainings are equally positive, for the patients. So with this, actually, we developed the algorithm that can complement, and I'm not saying that replicates, because of course, the pathologist assessment is a critical at this point, but complement in a high throughput the digital scoring. So with digital algorithm, we are able to completely replicate the TAP scoring and CPS scoring. And we actually did in a blind manner, a study with more than 100 samples that were included in this study, comparison between pathological evaluation and digital pathology. And in a good way, surprisingly, right? So we've seen quite concordant results. So which actually like was part of the validation of the algorithm. And again, every single work that we are doing, so we would like to focus not only on analytical component, but on the clinical component. So I do have quite a lot of confidence in terms of the performance of this algorithm. So it can be applied, of course, as a part of the clinical trial on exploratory level, but it has a lot of potential. So to summarize, and again, so make it like summary of the presentation that I just gave you in a very high level. So the digital pathology actually provides a very cost-efficient, high throughput analysis that actually combined pathologist expertise with potential AI assistance. So it supports collaboration, enables large-scale data research, which could be a really critical component when you have like multiple sites involved with the different capabilities to run the studies. And the high throughput is really required. And this is actually the solution for digital pathology endpoint to be introduced. So for translational and clinical benefits. So there are a lot of information that we can use for considering that the clinical validation is of course important, but translational science and translation digital pathology can be simply implemented as part of the clinical benefits for any biomarker identifications, for pattern recognitions, and it could be used as a potential diagnostical tool after appropriate, I would say, analytical and clinical validations workflow. So what is more exciting and then just using like the same slide, so you don't need to kind of reproduce some of the stainings or add additional slides using the same IHC slide that has been generated over the course of the staining. So you can do much more parameters in terms of the evaluation, like cell-to-cell interaction, neighborhood analysis, that it truly shows like the PD effect of your biomarker of interest that you used. And as one of the example of the tumor phenotyping, you can go much more in terms of characteristics of the tissue samples, and this could be considered as a potential criteria for randomization, stratification factor, or predictive biomarker for patient therapy outcome. So with this, I would like to thank you all for your attention. I also would like to take a moment to really acknowledge the Novartis Diagnostical Science and Partnership Group and Precision Medicine Clinical Innovation for like full support and endorsement of this work. And in case of any questions, I'm happy to take them either online or offline. And this is just information with the email address for a few associates from my team and myself, right? So thank you so much again for your time.
Thank you, Alexander. Really, really amazing work and presentation.
Thank you.
Okay, so the next speaker is Daria, CTO of Guardant Health, talking about the new wave of innovation in liquid biopsy. Daria.
Thank you very much, Asaf. Happy to join. Replace current share is what I'm being offered. So let's do that. And I'm hoping you can see my slides.
Yes.
Awesome. So good morning, afternoon, evening, depending where you are, for joining the multimodal dive that we have today at BladBack. I'm excited to continue the thread that you heard from Tuvik and Alexander on richer tumor phenotypes being inferred to improve patient treatment. We're seeing that wave coming through in tissue, digital pathology, and we're really dedicated to make sure liquid biopsies are continuing to innovate in the space and make tumor phenotype available from blood sample analysis. And so I'll focus my talk today on that aspect of what liquid can bring to the table. Clearly, we talked about this, and Tuvik stole 2 of my slides by now, so I'm really happy that we're thinking about it as a field together. Genomics alone doesn't answer everything that physicians need to treat their patients. And even though we enabled the revolution in precision oncology over a decade ago, we still have pretty far to go to maximize the benefit to the patients. Genomic mutations don't do that. So with our smart platform unifying genomics and epigenomics to reveal underlying biology at unprecedented scale, we're redefining how clinical decisions can be made. And we do believe that epigenomics does represent a new frontier, pun intended for Lauren's topic here, the next frontier here. Epigenomics signals obviously encode underlying tumor biology, much similar to the RNA profiles that Tuvik talked about. Yet these epigenomic signals are much more stable and robust in terms of assessment in a liquid biopsy sample than RNA is. And so that represents a clinically relevant avenue for expanding these insights as we think about patient care. I think most of the people around this table know what genomic and epigenomics is. Very simply, genomics tells us the hardware or the structure of the tumor. Epigenomics tells us the software layer on top of it that helps us know how the tumor operates or functions. And so by combining these two, we live in the world of richer inference over system level view of tumor genotype and tumor phenotype. And given, again, the richness of biology that is encoded by these signals, we can both detect and quantify the tumor based on the intensity of methylation that we measure in blood. I'm not going to talk about this much in the talk today, but we can also characterize and predict how the tumors are going to behave, assess their changes longitudinally under treatment pressure, and really read that tumor state in a blood sample through the lens of epigenomic regulation. How is this possible? This is enabled by the proprietary biochemistry and technology that we've built at Garden over the last 8 to 10 years that really is focused on capturing genomic and epigenomic data from a single sample without loss of signal in either of the two channels. In our evaluations against multiple off-the-shelf technologies, including bisulfite, as well as newer enzymatic methods for less destructive methylation readout, we continue to observe higher signal-to-noise characteristics in molecule capture, with superior retention of signaling molecules and effective depletion of the background. And so that approach, if you think about sort of the last many years of epigenomics, bisulfite with harsh treatment and other impacts in targeted sequencing data, really minimize the ability to use it in a clinical setting on scarce samples. We've solved that problem and can now measure genome-wide methylation profiles with reasonably high efficiency, not reasonably, but with as high efficiency as traditional genomic liquid biopsies do. And so that represents the window of opportunity here to harvest that signal. These platforms are now supporting all of the data that we obtain in our patient samples today when we run Garden 360 tests. So what that means in practical reality, that we've now accumulated close to 1 million profiles in our database from the years of testing that we've done, and probably 350,000 of epigenomic profiles, again, at the genome-wide scale. What that data allows us to do when placed into the context of real-world data accompanying these patients, including their response to therapeutics, what drug they were on, and history pre-blood draw and post-blood draw, is really high throughput discovery, multimodal signature development, and retrospective validation effort that database enables. So we're doing that discovery at roughly at 1 million patient scale, which is kind of a dream I've had from coming into the space of biotechnology 15 years ago, where we're limited to very small patient cohorts. So what does that mean? The data-driven innovation that is enabled by the platform collecting this data and patient testing that's occurring to collect that data at scale unlocks entirely new capability for liquid biopsy. They've been adopted widely for genomic biomarkers, but most phenotypic markers that define how the tumor functions are only accessible through histology, like you've seen in the previous talk, or other direct analysis of the tissue. That means all of the limitations of scant tissue amounts QNS apply, but most importantly, it limits the ability to assess that patient longitudinally over time as the disease progresses, as the disease evolves, and we've seen that the metastatic evolution of the tumor over time is really not, cannot be adequately treated unless you know what happens in the underlying tumor. So we are at a point where molecular and real-world data associated with this patient define a high-resolution map where patients using similar tumor characteristics cluster together. As our database grows, we can map these patients with higher resolution and direct patients to therapies with higher likelihood of response. By integrating these diverse data sources, we've validated several new features that are redefining how tumor phenotype can be assessed. A few of them are launched today. We've created features that recapitulate known tumor phenotypes, such as tumor site of origin and molecular tumor types like HR-positive status in breast cancer. And we're very excited about novel phenotypes that are currently in development and promise to identify more patients eligible for treatments or trials, or predict response for approved therapeutics and expand indications. Let me start with an example that makes a difference in the clinic today. While in some cases tumor origin is known at diagnosis, for many patients the disease has advanced so much that it's very time-consuming or nearly impossible to identify the origin. yet no one clearly changes the treatment pathway. We trained artificial intelligence and machine learning models to differentiate methylation patterns observed in blood and provide confidence rank predictions of the tumor origin. These predictions, with made at high confidence, parallel the accuracy that is afforded by direct tissue NGS methods based on RNA analysis. Another future exciting application of that is identification of secondary primaries that arise in surveillance of MRD patients. Being a clinical company, we are very driven by the outcome we create for patients and physicians. And this particular feature, as trivial as it sounds, has received very good feedback from the providers who are practicing medicine, not on the ivory tower of kind of hypotheticals, but on the clinical reality of how patient care is practiced today. In this example here, tissue NGS was QNS. IHC could not be used to conclusively type the tumor. Our results came back as 100% biliary cancer, and this leading physicians at a hospital is using that to define the treatment landscape for that patient. At a finer resolution, it's not only tumor origin, but also molecular type that defines optimal treatment, like a hormone receptor status in breast cancer. It has been known for a very long time that these types are critical to putting patients on the right treatment, but yet nearly impossible to know dynamically during treatment how to ascertain changes in that phenotype with tools due to lack of non-invasive tools for doing so. We're able to demonstrate that we can match these molecular types to their associated epigenomic profiles and identify clusters of profiles that map, for example, to HR positive or negative status. Now, applying this capability to patient samples tested over time during treatment, we're able to offer longitudinal assessment of the molecular types and identify any type transitions like HR negative in this example at the latest time point. So today, physicians finally have a tool that's available clinically and validated with relevant patient clinical samples that assesses breast cancer subtype transitions over time, as well as lung cancer subtype transitions. And this is just opening the door to the concept of tumor subtyping using epigenomic information. In our next challenge, we worked on increasing clinical sensitivity for traditional genomic biomarkers with the goal of identifying more patients eligible for treatment. Despite the success of liquid biopsy, we all know around this table that there are classes of biomarkers that present challenges for detection, especially when the tumor levels in circulations are low. We're discovering that many of these genomic drivers are associated with distinct methylation profiles and can be rescued by bringing methylation data into the algorithms. Let me show you two case studies. In the first one, we evaluated ALK fusion. This biomarker is associated with therapies that deliver unprecedented 80% plus response rates. So any missed treatment opportunity is a miss for that patient. Using our database of over 200,000 individuals, profiled longitudinally, and providing real-world evidence data, we derived a high accuracy signature that is associated with ALK and demonstrated that nearly 40% of samples where ALK fusion was missed were recovered by a positive call of the epigenomic surrogate signature. In another example, we evaluated MTAP deletion, another marker that's traditionally hard for liquid biopsies that are just using genomic information. And this is an emerging pan-cancer biomarker, which is currently used in over a dozen clinical trials. And methylome signature in this case identified 2.4 times more patients than genotyping alone. These features will meaningfully increase the utility of liquid biopsy testing and offer clear advantage over genotyping alone. Finally, our most exciting new applications are being developed to address a major gap in current precision medicine, where existing biomarkers are only partially predictive of response. Richer information is needed. With the data density increases, increasing, as well as longitudinal follow-up data maturing in our databases, we are uncovering methylation signatures, separating the groups of responders and non-responders. We've demonstrated feasibility of this approach across multiple therapeutic classes and 3 major cancer types. Let me walk you through that data. Starting with lung cancer, we've developed a multimodal signature of response to immuno-oncology therapy. On the left, I'm showing ability of the current TMB biomarker to stratify this group of patients derived from our real-world evidence cohorts. This is tumor mutational burden. And when you combine TMB with promoter methylation status across multiple biomarkers, you are able to achieve much better stratification of the patient response. In colorectal cancer, we've evaluated anti-EGFR therapies. Here, the best you can do clinically today for cetuximab is sidedness of the tumor. Left side gets one treatment, right side gets the other. Guess what? By incorporating this true biology of the tumor into the decision-making process, we're able to better stratify the patients than the sidedness or other clinical indicators do on their own today. We're showing patients on the right with right-sided tumors or a subset of those patients actually responding to that treatment. Finally, in breast cancer, ESR1 mutations are established biomarkers of resistance that support transitioning from aromatase inhibitors to SERD-based endocrine therapy in AR-positive patients. However, some patients who lack detectable ESR1 mutations show benefit from SERDs without a reliable indicator of who would benefit and who wouldn't. We developed a methylation-based AHR activity score to epigenetically characterize the molecular subtype and demonstrated that in ESR1 negative population, that activity score meaningfully differentiates response to SERDs, offering yet another opportunity to refine the meaning of precision medicine in this population of patients and go beyond ESR1 mutations as the stratification biomarker. All of this goes to the original statement that I think we're all ready to make today in this panel, where the data and science are converging to provide new dimensions to treatment selection. As we grow our databases from 10,000 to 100,000 to million patients, the information richness allows us to improve that stratification, and we are very dedicated to that cycle of incremental improvement work that is driven by the data richness and more refined look at patient tumor evolution over time, making that the basis for treatment decisions that are made. It provides opportunities both on the clinical side and on the translational side to work together in identifying non-responder patients and understanding their underlying tumor biology. What are the options to intervene therapeutically? Because without that, clearly, we're underserving what we can do with this data. And so looking at response predictions in previously failed trials, as well as ongoing trials, enhancing patient identification for enrollment, further molecular subtyping, and adverse event predictions are examples of where we're working with partners to refine approach to precision medicine. With that, I am really very grateful to a very large team at Guardant behind this development effort. We've been dedicated to bringing out these signals for a number of years, and for people who are practicing in the field, the translation from idea to a clinically validated tool is a multi-year process, and we're very happy to see convergence of that, seeing both with the work we've done here inside the walls and the works happening outside, with so many of these biomarkers crossing the translational relevance into clinical applications. So really looking forward to how many patients we can impact as a result of this work together. Happy to discuss with others during this panel discussion. Thank you.
Thank you, Daria. Really amazing demonstration of the power of multi-omic integration within liquid biopsy. The next speaker is going to be Timothy Texter, Executive Medical Director at Tempus AI. He will be speaking about clinical integration of multi-omics biomarkers in endometrial cancer. Tim.
It's off. Let me make this full screen. So still, just want to double check. Is this still full screen for everybody? Perfect. All right, good stuff. Right. Okay, so yeah, I'll go back to the template slide real quick. So yeah, I'm gonna speak today about a clinical implementation strategy of how you use multi-omics and using endometrial cancer as kind of a case study. I think it's a good example that highlights a bunch of different use cases. I'm a pathologist by training. I've been at Tempus for 10 years. And during the last 5 years, I've been kind of specifically focusing on building, developing, and launching a lot of these multi-omic algorithms that I'm gonna chat about today. So yeah, overview of what we'll talk about. I'll give a bit of a background on the company, and then discuss some of our lab testing services. And then we'll kind of pivot and focus specifically on endometrial cancer, which is a good example of where these algorithms kind of come together in a cohesive strategy. And hopefully I didn't pack too much into this talk. We'll see. We'll see if we can get through everything. So just a bit of a background on Tempus. Since the beginning, the company was always designed to be this kind of operating system, deriving clinical insights from all the different types of, medical data available. And that's really, that mission's really central to the products that we'll talk about today. We've been operating at scale for a long time now. We're working with, majority of the US health systems and most biopharma companies. The data that we're working with is kind of inherently multimodal and growing at a pretty large scale. We have a big team. This actually doesn't include our recent friends from Ambry that have joined the team. So we're excited about that, doing more hereditary testing in oncology and cardio. This is a little bit more on partners, which we already kind of touched on. Oncology has always been really it was the beginning, the core, focus of the company. That's true today as it was, 10 years ago. But I think it's worth noting, we have been trying to apply, our platform and technologies to other disease areas. I think the one that's kind of specific to call out is cardiology, where we were doing some pretty exciting work, using EKGs to predict structural heart disease, predict AFib, which got 510 clearance last year. And then radiology is really kind of permeating a bunch of disease areas, oncology and cardiovascular. I think there's a lot more interesting applications that'll come in the future of that. So the company's kind of generally structured in these sort of 3 macro buckets of genomics, which is all of the lab testing services and molecular laboratory services, data, which is predominantly life sciences, and then apps, which is really where a lot of these like nascent AI-driven, clinical decision support technologies are housed. The big focus today, at least especially for what I was asked to do, is kind of hit on this idea of multi-omics and how it applies to clinical medicine. And I think it's worth calling out that, all the things, at least at Tempus, that we're enabled to do are really possible because of this infrastructure that we've been building that supports all of the different types of medical data available from, radiology, the molecular pathology lab, including, our in-house panels, other testing, the clinical lab with clinical data, anatomic histology with, scan histology slides. So all of that together is really what's kind of driving development and commercialization of the different tests that we have available. This slide is just kind of highlighting, the value of the different, products available, starting on the left from the different lab tests that we have, which I'll get into in a minute, and then kind of progressing farther to the right and increasing value of these, what I mentioned kind of before, some of these app-related products that are these clinical decision support tools like Tempus Next. But I did want to call out, that the y-axis also could represent, the level of integration, because really like the value that you can deliver is a function of, the amount of data and the level of integration that you have. It's really kind of like the ceiling of precision medicine in some ways is a function of that, scale of the data and ability to access it. I think that's kind of important to consider as we move through. As I mentioned, yeah, genomics is the kind of the beginning and still the focus of the company. These are all the different lab offerings, and there's probably some stuff that's not on here. I won't get into the details of all these different tests, but really kind of the workhorse of the company is XT. That's our targeted sequencing DNA assay, and XR is whole transcriptome RNA, and then we also have XF is liquid biopsy, and then we'll hit some of these algorithms later. This is another look at those products, but from the kind of patient journey perspective. And I just wanted to put this slide in there because it nicely highlights, sort of the goal of, we really want to try to offer, a comprehensive, like sort of lab services package that can, inform decision making all the way from, diagnosis, early stage disease management up through late stage disease. And today we'll kind of focus explicitly with endometrial cancer, which is a nice disease example to kind of dig into a few of these different products that have sort of that are kind of inherently like multi-omic based and kind of stretch across a bunch of technologies that we're working on. So I think should be pretty good. But before we do that, we should do a little bit of a deep dive and background on endometrial cancer management, which is a pretty complex space. I got to learn this firsthand a few years ago as we started exploring algorithm opportunities in this space. I'll do my best to kind of condense what I've learned and sort of what I think sort of has evolved over the last 20 years. So you basically have three different levers you can pull in this disease for treatments. There's systemic therapy, like chemo for controlling distant recurrence, external beam radiation for controlling pelvic recurrence. That also theoretically could control distant recurrence by wiping out pelvic micrometastases. And then there's brachytherapy for controlling vaginal recurrence. And so maybe back like 20 years ago, this disease was predominantly, managed through, anatomic staging. But then over, the last, maybe decade or so, a lot of researchers have been through the Gynecologic Oncology Group, which is GOG, that's US based and dominated by surgeons. Then there's Portec in Europe, that's mostly radiation oncologists. They've been working on retrospective analyses of past clinical trials, trying to understand how to integrate different clinical and pathologic features. That has now morphed into this sort of risk schema that you see at the bottom with low risk, low intermediate, high intermediate, and high risk. And so it's a little bit convoluted of like but there's different features and weightings for how these are used. And it's brought some good specificity to the disease, but there's still some gaps in terms of, as you can see here, there's kind of a big spread of options for high intermediate risk. There's a general acceptance that it'd be great. There's probably high intermediate risk patients that need chemo, and then there's likely high risk patients that likely could be de-escalated. It's also a lot of interesting nuances between the surgeons and the radiation oncologists and their opinions, but we don't have time to get into that. So one thing to highlight is, the field has gone through a big change with the advent of, or the integration of molecular stratification. So there's basically 4 different groups in endometrial cancer. There's T53 for serous-like histology. There's this POL-E hypermutated phenotype, MSI, and then there's this like other group that's called NSMP. This is like non-specific molecular group. And what these investigators found is that the POL-E patients do very well regardless of histology. They have like exceptional survival. And the P53 or serous-like high copy number, those patients do very poorly. And so recently the FIGO staging was actually updated to include molecular classification. And so this was like a huge deal in the field, and clinicians are trying to figure out how to integrate it into academic labs, because POL-E testing is really like not standard at all. It's actually pretty complicated to implement. and then there's also still this kind of open bucket of like, what do you do with the NSMP and MSI patients? They have indeterminate prognosis. Like what does that mean for distant recurrence? And this is what got us excited about this space. We were building some RNA signatures, looking at trying to differentiate risk of distant recurrence. But during that time, this really cool digital pathology algorithm came out of the University of Leiden that was led by a pathologist named Shaling Basse and an AI scientist named Sarah Valinsky. I apologize. I should have put an H in Sarah's name. Let's correct that. And so what this algorithm is doing is it's using clinical data plus the digital pathology scan to predict distant recurrence. Plus, it can also inform outcomes on adjuvant therapy, adjuvant chemotherapy. And so we embarked on a licensing arrangement with this group and are looking to launch this test as part of our offerings in DigPath. And so this is we'll do a quick summary of the data there, but this is highlighting its ability to predict distant recurrence and stratify across the 3 different risk groups of low, intermediate, high predicted by Hector. And then what's really cool is they also have data from the PORTEC-3 randomized controlled trial, which was comparing EBRT, so radiation therapy, plus or minus chemo. And in the Hector high-risk group, it actually was stratifying and predicting patients that could benefit from chemotherapy, which is a huge question in the field. And so, yeah, there's more to come on that, but this is, potentially a very valuable tool. And, there's a lot of molecular early stage disease, predictors, across a lot of other cancers. And so endometrial cancer is kind of waiting for something like this to happen. To tie it back to liquid biopsy, since that is sort of the nature of blood pack, I thought I'd touch on MRD briefly. Right now, this is not standard of care by any means in endometrial cancer. Natera is doing kind of the most work, I would say, in this space. They had some really cool data, I think it was last year, demonstrating the potential value in a retrospective study, and demonstrating that, yeah, it's independently a pretty strong prognostic factor for recurrence. Tempus has on the right, I just want to highlight like a few different methods that we use. We have methylation based assay and targeted DNA. That's a tumor agnostic approach that we use in colorectal cancer. And then we also have this partnership with Personalis for tumor informed MRD testing that's offered in NSCLC, CRC, and breast. So I think MRD and endometrial cancer is definitely going to come. I think it's more just a question of when and exactly how it'll be implemented. So I think there'll be more to come on that in the future with additional clinical trials. Next, we'll transition to emerging biomarkers in late stage disease. So we'll get into more of the kind of standard treatment selection that I think we're all used to, which highlights, a bunch of other different multi-omic strategies. And so, currently in the approved bucket, it's limited to, really just MSI status and using MSI status for the approvals of checkpoint inhibitors such as dostarlimab, pembro, lenvatinib, or pembro by itself, and durva. But I think this is kind of an interesting space because there is, I think kind of an interesting, opportunity that's starting to exist at kind of the confluence, I would say, of the, DNA or the DNA damage space and the ADC space, where you have a lot of trials going on evaluating topoisomerase inhibitors that are kind of like in that DNA damage space, along with the targeted DNA damage drugs such as PARP inhibitors, which a lot of us are familiar with, but also things like ATR inhibitors as well that are being trialed out in endometrial cancer. And so it's kind of this like whole world of I would say DNA damage. That's a really interesting place right now for where multi-omics can expand, because right now it's primarily just based on MSI testing for IO, and then alongside that IO point as well is there was some recent data with the Keynote 868 trial where there was like positive PFS data in patients that were mismatch repair proficient. So there's been kind of a debate in the field where there's a lot of clinicians that are interested in, what are the patients that might actually, benefit from, expanded use of IO outside of the pure, mismatch repair positive patients. And so, there's a lot, I think that's also kind of a space where, there's a lot of activity and a lot of interest in trying to, possibly expand usage and what biomarkers can help inform that space. This is also not a completely exhaustive list. There's also some interesting strategies in the mTOR and PIK3A pathway. But I thought this was generally helpful, and also just highlighting sort of each of the different modalities between like DNA, RNA, other like HRD. There's lots of different approaches. So I'll highlight two different algos that we have, one in the IO space and one in the HRD space before we wrap up, that kind of highlights sort of a multi-omic approach that we use. So IPS stands for Immune Profile Signature. We launched this almost exactly a year ago at SITC last year. And so this test was, designed to improve upon. Is that 10 minutes,
sorry.
Doris?
Do we have 1 minute left?
Okay, great. I'll go fast. Cool. So yeah, this was improve upon the current IO biomarkers. This is the high level sort of example of the machine learning framework where we used existing IO biomarkers to then distill it down to this final signature, and we used sort of a large data set of ICI treated patients. And we're working to kind of expand the clinical development space of that assay to figure out additional indications of where it could be useful. And then finally, just to wrap up, as I mentioned, HRD has been something we've been interested in a while. We developed an RNA strategy in this because of the limitations of just DNA-based models for non-ovarian and breast cancer. And we have recently been working on an update of that assay. And this was some preliminary data showing the gene features that are and what pathways are enriched. So more to come on that in the future. But we're excited about that, especially as the space matures in endometrial cancer. So yeah, just to wrap up, I think the goal was to just highlight, I went pretty fast, but to highlight, there's a lot of different tools that can be beneficial as you go across, the patient journey, depending on what the indication is. So thanks.
Okay, thank you, Tim, and thank you to all of the speakers. Really exciting work in progress. I'm going to hand it out to Jim now to lead our panel. So, Jim.
Thank you, Asaf. Good afternoon, everyone. Speakers, that was an outstanding setup for the next portion of our seminar, which is the panel Q&A. It's my pleasure to be moderating today's panel discussion. My name is Jim Gotzi. I'm a proud original member of BloodPAC, and I've served as a scientific co-chair for the past 9 years. So as is our tradition, we've assembled a panel of subject matter experts representing a diverse set of skills and experiences to address a number of questions with regard to the many challenges to be overcome as new tests that are based on integrated multimodal data attempt to make the way to the clinic, and doing so fulfilling one of the primary objectives of the BloodPAC's mission statement. So let's start by kicking off the panel discussion by having each of the panel members briefly introduce themselves. So let's go back a slide, and we'll start with Ben.
Hi, everybody. Nice to be here. My name is Ben Glass. I work at PathAI, leading our product AI teams and translational research teams, and have been working at PathAI for around 10 years now, developing AI tools for digital pathology. So really excited to be here.
Kareem.
Hi, this is Kareem Mauser, and I'm head of data and AI and clinical development at Novartis for cardiorenal metabolism and neuroscience. I've been in the quantitative field in pharma, biotech, and global health for over 25 years, and delighted to be here and this exciting opportunity to talk about new frontiers in therapy selection for oncology.
Matt.
Hi, all. Thanks so much for inviting me. My name is Matt Eaton. I lead the computational biology and bioinformatics group at Proceed Biosciences, where we're using liquid biopsy to predict disease gene expression.
And Orly.
Hi, everyone. Excited to be a new member of the Blood Pack. Thanks to Asaf. My name is Orly Alter. I'm Ustar Associate Professor of Bioengineering and Human Genetics at the Scientific Computing and Imaging Institute and the Hands-on Cancer Institute at the University of Utah. I'm a scientific advisory board member of the NCI DOE Cancer Moonshot Collaboration on Predictive Oncology and the inventor of the IgG. I'm here as the CSO and a co-founder of our university spin-off, Prism AI Therapeutics, where we have a platform for your drug and biomarker development with our quantum mechanics-based artificial intelligence and machine learning, or AI/ML. Thanks for having me.
Very good. So before jumping into the first question, in an attempt to ensure that we get through the majority of our pre-planned questions, I will identify a single responder amongst the panelists to respond to your questions. At 10 minutes before the hour, we'll pause, take audience questions. If we have no audience questions, we'll simply recycle back to the pre-planned questions. So why don't we bring up that first question, which goes to Orly. What are the opportunities for integrated multimodal data to expand access to precision medicine for patients beyond traditional genotyping modalities? Orly.
Thanks, Jim. Thanks, everyone. So prediction in medicine remains limited, and in my mind, this is the great challenge as well as the greatest opportunity. Integrated multimodal data can significantly improve upon prediction in medicine, and in this way, also upon the understanding and control of disease. And I'm fairly certain everybody would agree. You see, today we unfortunately experience an increase in drug failure rate from 90% to possibly 95%. That's while the development of targeted therapies that work, say, against one DNA mutated gene or one overexpressed protein reached an all-time high. And this is because biotech companies and healthcare systems still do not use their own data, really your own data, from preclinical and early trials to post-market follow-ups to make predictions. For example, to predict which patients would live longer thanks to a drug. And as clinical studies demonstrated, a measurement of the one gene mutation or the one protein overexpression does not answer these questions, not even in the case of a targeted therapy. And like geneticists working with model organisms showed, the activity of a drug, even one that targets one gene or one protein, depends on the whole molecular, really the whole multi-omic background. So the greatest opportunity for integrated multimodal data, not just any data, but your real world data, is to improve upon prediction and in this way also upon the understanding and control of disease. But you say, my data are small cohort, noisy, and high dimensional. So at Prism AI Therapeutics, we have developed our quantum mechanics-based multi-tensor AI/ML to overcome these challenges. And with our partners, we have shown in clinical trials and CRISPR experiments that our AI/ML works. It is uniquely able to discover in your data predictors that validate. So we ran, say, 2 use cases that were published at AACR and ASCO this past year, for an example. And in both cases, our platform discovered in an unsupervised manner whole transcriptome predictors of a patient's overall survival and objective response to a targeted therapy from real-world clinical trial data. In the first case, the data were from a genetic clinical study of the immunotherapy drug atezolizumab in patients enrolled with advanced or metastatic bladder cancer with high PD-L1 expression levels, and we heard about PD-L1 earlier today. And in a second case, the data from only 19 patients were from the tumor-agnostic NCI-match subprotocol age study, where patients presenting with tumors with BRAF mutations were treated with the Novartis combination therapy of the dabrafenib and trametinib. This use case and our follow-up ongoing work with the NCI Match is in collaboration with a fantastic Dr. Lindsay Harris, Associate Director of the NCI Cancer Diagnosis Program, whom you all know from her work with you at the Blood Pack too. So we're excited about that. In both cases, the predictors clearly outperformed the traditional one gene BRAF mutation or one protein PDL1 overexpression. In the case of the genetic trial, the predictors also outperformed the best reported immunotherapy indicator, that's tumor mutation burden. They were more accurate with greater concordance indices and with greater conferred hazards than tumor mutation burden. And in both cases, our proprietary algorithms also validated those predictors in publicly available data from TCGA. It was in the general population and regardless of BRAF mutation or PDL1 overexpression, and extended the phenotypes to larger percentages of the patients. In the case of the NCI match trial, the validation was on 3 different tumor types, including melanoma, which was not even represented among the 19 patients in the discovery set, extending the phenotype additionally across indications. And in both cases, our interpretations or black boxing of the predictors identified mechanisms of resistance as well as drug targets to sensitize the tumors to the treatment additionally, essentially extending the genotypes. In the NCI match trial, these are in agreement with the known dependence of a patient's response or the tumor's histology, and specifically the confirmed response of colorectal cancer patients to the targeting of BRAF together with EGFR. So to summarize, with proper AI/ML, integrated multimodal data can significantly improve upon prediction in medicine, and in this way also upon the understanding and control of disease.
Thank you, Orly. So question number 2 goes out to Matt. So what are the opportunities for integrated multimodal data to improve drug development and patient stratification in clinical trials?
Yeah, thanks, Jim. So I think the real opportunity here is to make drug development more predictable and interpretable. So, you know, in oncology especially, and just to sort of echo Orly here, you know, the probability of success for new drugs hasn't really budged in 20 years. Although we've made huge scientific advances like tumor transcriptional profiling and huge databases like TCGA. And, you know, with R&D costs climbing, we've got to find ways to get effective medicines to the right patients with the same or sometimes even smaller budgets. And so the opportunity, as we see it at Proceed, is expanding access to directly interpretable transcriptional biology at scale. We need to understand in real biological terms why patients respond, why they don't, and why some patients who at first respond eventually stop. And that understanding drives better patient selection, smarter combinations, sequencing strategies that actually boost success rates. So, you know, for example, what we're doing at Proceed is to bring RNA-seq-like readouts of tumor and immune biology into a liquid biopsy format. So from a small volume of plasma, we can capture the expression level biology that drives response. It's accessible, it scales, it fits right into existing trial workflows. We're using plasma draw protocols that people are already familiar with. And we're seeing it sharpen patient selection and even extend the reach of some therapies. So for example, in breast cancer, we've seen patterns of pathway dependency that line up really well with tissue RNA-seq across more than 200 patients in 3 independent cohorts. Those differences track strongly with progression-free survival. That's data that we'll be presenting at SABCS this year, but it's, you know, it's a great example of how liquid expression biomarkers can connect mechanism to outcome. So, you know, I think the bigger picture here is, you know, integrating multi-omic data, bringing in, you know, liquid ways to measure expression along with things like CGP and digital path. That lets us link dynamic biology, you know, right to clinical outcomes, and sort of, you know, make use of all the amazing research that we've done in the last 20 years to understand disease biology. And that's how we get better patient stratification and smarter, higher success clinical trials.
Thank you, Matt. So question number 3, Dr. Kim. So what types of indications are intrinsically best captured by the different types of multi-omic modalities? How do you think about the trade-off between the ability or perform an assay to resolve the biomarker of interest versus clinical feasibility and throughput requirements? Kim.
Nice. Yeah, I think, you know, when I started building, I guess, algorithms maybe 5 years ago, kind of approached it the way a lot of people do, where like you have a technology that you like, and you kind of, look at the world, you're a hammer looking for nails. In our case, it was RNA, and we were really just kind of pushing the limits of like, what can we do with that? But as we kind of got more sophisticated and launched more and more tests, I mean, what you really want to do is start with the clinical target product profile, really try to figure out like what is the performance that you're trying to hit for biomarker resolution, and also what is the clinical context, what kind of constraints are there that are relevant, and then you work backwards to the technology. And so, that's kind of what I was hoping to emphasize a bit today in the presentation of like, what's the right tool for the job. I think like early stage disease is a good example of this. There's molecular assays have been used, for a long time in that space. Like Oncotype DX is the, the OG, right? It's the one. But what's going on now, there's a lot more, tech development of DigPath in, a lot of other diseases and across early stage. And, the performance of those tests is like as good as like a molecular based assay. And so it also has the advantage of like throughput, like turnaround time is much better. You don't have to do sequencing, so you can deliver results on more patients. So in that case, it's like you have like technology equivalency in performance, and it's much better from a workflow perspective. So it's like, why would you use anything other than DigPath for early stage disease versus, something like predicting cancer type, which was our first algo, was tumor origin. I'll use DigPath as like another example. Like those DigPath predictors for cancer of unknown primary, are kind of have a inherently kind of low ceiling of performance. That's not quite good enough for actually like using them if you're really trying to do, cup diagnosis legitimately, versus, something like RNA-seq or methylation, can get you that extra resolution. So I think I've been kind of appreciating more and more, like you got to be practical about multi-omics. Like don't like overdo it. Like don't use technologies if you don't have to. Like you don't need to do proteomics on everybody, right? So I don't know. To me, it's kind of like trying to start with sort of the clinical problem, work backwards has been helpful.
Very good. Thank you, Tim. So let's advance the slide to the next slide, please. And for the next question, it goes to Kareem. So question 4, what are some approaches that multimodal AI model developers should use to ensure model interpretability and explain AI decision-making processes for various stakeholder groups such as clinicians, patients, regulators? And basically, how do we get past the black box nature of these types of models? Kareem.
Thanks for the question, Jim. So I think to ensure interpretability and transparency in a multimodal AI context, especially for diverse stakeholders like clinicians, patients, regulators, we need a structured approach to ensure that this multidisciplinary nature and the voice of these stakeholders are heard. Model development should start with a clear question in mind. What are we solving for? And unpacking that question in the form of a target product profile for the multimodal AI models, which is, a framework that we're used to in drug development so that all the teams are aligned on what we're doing. Now, this involves integrating best practices across model-based clinician, drug development disciplines, and ensuring we have a transparent operating model with clearly defined development stages. I would say there's 3 stages. So the early stage I alluded to a little bit before is involving clinicians, the patients, and their voice and regulators in building this target product profile. What would the product look like? What would need to be incorporated? What this does is ensure that the context for interpretability and explainability is framed appropriately. Now, effective communication and translation across these disciplines at this early stage is very important. The flexibility to be able to understand how can the technology be adapted to specific questions and needs for each of this group. That's why the role of translators and cross-disciplinary connectors on the team becomes invaluable, ensuring that the model development and explanations are accessible and meaningful to all stakeholders. Then comes the second stage, and that's really engineering the system, right? What are the inputs and outputs? Are they robust to what our understanding of the science, the clinical aspects, and that helps us preserve the robust data-driven model development. Achieving the right balance is another thing. When you think about explainability, there is a balance between explainability and model performance. So what is the right balance of that? And that's where the context becomes so critical. And as recently highlighted in an FDA workshop not that long ago, that level of explainability depends on the application and its influence and potential impact. Finally, I would say the last step is, now that we have a prototype or a developed tool, benchmarking studies that compare model behavior to, is the output what we expect? Is it consistent with practice? So that helps us understand what are the transparent drivers of these model predictions by identifying which features most influence the model, ensure they align with the clinical experience. We can build trust with these stakeholders and address both anticipated and accepted behaviors within this context of use. This supports not just the upfront engineering, but also ongoing assessment throughout this model lifecycle that needs to be defined very well upfront, as I mentioned, reinforcing interpretability and the context under which what we mean by explainability and interpretability. And finally, I would say, there's clear guidance from the FDA on good machine learning best practices. These provide really good guardrails to follow through this whole cycle. Thank you for your question.
So Kareem, I love the building trust aspect of your response. I think that's going to be absolutely critical. All right, next question, back to Matt. Question 5, how can we integrate and discover insights across multimodal measurements, liquid biopsy, digital path, et cetera, and biological samples in order to build actual products that's bridging the gap between discovery, validation, all the way to clinical tools? Matt, you're on mute.
Sorry. Yeah, thanks, Jim. Great question. I think, the two keys here for building multi-omic products are, interpretability and deployment. So the less interpretable the models are, the more you have to lean on massive cohorts just to prove that the signal's real. And that's a heavy lift. It slows everything down. I think what we really need are platforms that give us insights that we can reason about. Ones that tie directly back to, known biology and pathways. And that way validation becomes faster, cleaner, it's easier to trust, regulators like it. And that scaling function, as a function of samples collected that Tim showed in his talk, I think it becomes way less steep and more approachable if, sort of the underlying model is really sort of biologically clear. And then the second challenge is translation, taking something that works beautifully in discovery and making it usable in the real world. Most care decisions don't happen at big academic centers, right? They happen in the community. So if a multi-omic test can't be delivered through the same channels or economics, right, as the tools clinicians already use, then it's not going to scale. So, these two things have been a big focus for us here at Proceed. We're building a platform that's inherently interpretable. We're resolving gene and pathway expression from a familiar analyte, which is circulating tumor DNA and its associated chromatin. ctDNA is already being collected and used every day to guide in therapy and disease. So we're meeting clinicians sort of where they already are. And that makes the translation to the clinic a lot more direct. We're also helping translational groups bring the last 20 years of RNA-seq research into clinical trials, baseline on treatment, resistance, and meeting those teams where they already work too. So I think what's really exciting is that by anchoring to something familiar and interpretable like that, you can start layering on other data, right? Pathology, mutations, imaging. You can build this rich multi-omic view of biology without creating a black box. So again, bringing it back to sort of this question of interpretability. So that's how we kind of see moving from discovery to real products. It's by designing for interpretability and deployability from day one.
Thank you, Matt. Next question is to Ben, as question 6, the need for continuous learning and updating of AI models to keep pace with evolving medical knowledge and diagnostic standards presents ongoing challenges for regulators. How do you see this challenge being addressed? Ben?
Yeah, thanks for the question. It's a good one, and one that's extremely important for the field of medical AI right now. And to start, we need to kind of think about the total life cycle of one of these AI products, which is something that at our company, Path AI, we've been doing a lot of, is sort of development of these tools with an eye on submitting them to the FDA. So to start, AI products need to be developed under the good machine learning practices that are, shared by the FDA. So there's a lot of good guidance there that we can use to understand the robust testing paradigms that we need to think about, transparency on how models were developed, including the training sets and assurance of data quality, and then the pre-definition of our validation data sets and how we approach validation. While this initial product that's approved via the regulators will be a locked model that follows these good machine learning practices, we're moving to a new strategy that several folks have already applied, which is using this predetermined change control plan, or other words called the PCCP, which is a really great process that you can use now at the FDA. It's essentially a plan for future changes that the AI manufacturer will submit when they submit their device for approval. So they'll sort of align on what that PCCP looks like with the regulators before the device is approved. In that PCCP, what we do is we focus on creating alignment on the product development and validation and post marketing plan. What we really align on are sort of the what of any modifications that we'll make with the products. And that can include, modifying the model itself. So we could align on the modification that, we'll retrain the model at some cadence with verified data from source A, B, and C. How we would do that, so the how of the changes, which would include modification protocols that us as manufacturers will use to develop, validate, and implement those changes in a safe and effective way. And then any impact of those changes, so describe any risks related to those changes and how us as AI manufacturers for these medical devices will ensure that the AI remains safe and effective after making changes. So in summary, following these good machine learning practices combined with robust post-marketing surveillance monitoring and using this PCCB pathway gives manufacturers a way of allowing for these incremental advances to products to ensure that we're making these really, great changes to the products based on evolving medical knowledge and diagnostics, and ensuring that our AI is still safe and effective for patient care and what its original intended use was.
Thank you, Ben. Next slide, please. So Daria, we have Tim. Okay, Tim is going to take question 7. What opportunities exist using liquid biopsy as the longitudinal layer in multimodal data that can inform a wide range of clinical management, such as monitoring, treatment, decision-making, adverse event prediction? Tim.
Thanks. Yeah, I think Daria probably hit this more than I did, but yeah, I think, I showed a little bit in the patient journey slide. The methylation-based assays and also targeted DNA can be used, in the early stage disease area for, monitoring recurrence, and assessing early recurrence. And then in, the later stage disease area, for treatment selection, I think that it's pretty interesting, as some of these, mechanisms and biomarkers have gotten more sophisticated, like HRD, for instance, in like prostate, let's say, that's an indication where like getting a biopsy could be pretty challenging, like in the later stage disease setting. And it's something like, doing a methylation-based assay might be like by far the best approach there. So I think, there's opportunities for liquid biopsy to become, become more and more useful in some of these, like, some of these specific mechanisms. I think on the adverse events side of the equation, which I believe you mentioned as well, That one, I think there's some interest. We had some interest a little while back, especially with like IO, for some of the, more severe adverse events. I think that one's tricky, to Matt's point around like, is there a test that's actually going to even be used that you could use to kind of ascertain, said event that you're trying to assess. In that world, just CBC plus like clinical data points might be the only thing available versus, some of the, immune profiling assays that you could theoretically do, but might be challenging. So I think it depends a little bit. I think like there's tons of opportunities, in some of these disease areas, and then there's some other ones that might be a bit of a challenge just for like, whether or not like a test is actually going to be available or not.
Thank you, Tim. Question 8 goes out to Alex. Alex, give me like 2 minutes on this. What are the key challenges and opportunities in implementing digital algorithms for PD-L1 scoring and other diagnostic assays in routine clinical practice?
Yeah, I think I will just gonna focus on mostly on challenges because they're like the same question I've seen on the chat. So there are multiple challenges, I would say, right? So that we are facing right now. First of all, there are like some sort of difference between what diagnostical assay has been applied. So, and we've seen quite a lot of shift to LDT test, lab developed test, to be implemented. And of course, like the scoring methods might be impacted by that, right? So that's kind of harmonization between the different diagnostical tools will be critical. So there are like technical factors, of course, affecting accuracy. So that needs to be addressed as well. And most importantly, and I think that's what was mentioned before, the part of the clinical validation, right, is a really important aspects, to make sure that the algorithm is validated, robust enough, and can be used across multiple sites. So this is like a main challenges. And of course, I should mention about regulatory approval, right? So there are like a lot of hesitance to really make it as a FDA approved and then or cleared, right? So that's a lot of speculation around what's the next step is going to be.
Thank you, Alex. And then Tuvik, final question. How can liquid biopsy tests expand therapy selection for patients who cannot undergo invasive tissue biopsies or whose tumors are difficult to sample?
Thanks, Jim, for the questions. It's a pretty broad one. And I would like to divide the answer into two. So first, there's a question of what do we want liquid biopsies to do in general? Do we see it as a substitute for the solid tissue biopsies, or do we see it, as most of the discussion here today revolve around, as an addition to it, as a complementary view of the tumor? And the second related question is, how are we actually going to use it? What types of assays, what types of inputs can we derive from the liquid biopsies that can help us get the answer to what treatment would be best for a patient? So regarding the first question, the main distinction that I would like to offer is between the liquid biopsy as a primary indication of treatment choice versus a monitoring tool to adjust a treatment that's already given. I think these are two very different scenarios, and they're both very, very important. My personal belief is that the second is a lower hanging fruit right now in many ways. And there's a lot that can be done to improve it. So specifically, our interest is really focused on looking at gene expression patterns, or more generally, activity patterns in the tumor and in its surrounding, including the immune surrounding, as a way of better understanding what's going on there with the tumor and how it would respond to various therapies. And with that, the challenge of using liquid biopsies is that if you're looking at DNA, if you're looking at the mutations, you can get a pretty straightforward readout from liquid biopsies, and it would be consistent. With sequencing RNA, for example, to get readouts about expression, it becomes way more tricky. It's more tricky because RNA sequencing is a tricky assay, so it's not absolute in nature. You have to have very good reference to interpret it, et cetera, et cetera. And it's also tricky because it's harder to identify what part of the RNA readings that you're getting is actually coming from the tumor. It's much, much harder than with DNA. So I think the first stage, if we want to go in that direction and really get a better understanding of activity patterns in the tumor, is to look at the whole variety of different assays that are out there. You can do the straightforward RNA sequencing from CDCs. You can do RNA sequencing from cell-free RNA that's in exosomes or protein-protected RNA. But you can go in the direction of ChIP-seq. You can go and look at fragmentomics as a way of measuring expression. And you can even go so far as say, let's just sequence PBMCs, because we already know there's a body of work that shows that sequencing RNA from PBMCs teaches us about the interaction between the immune system and the tumor. And in my opinion, we still need way broader exploration of all of these assays. We need massive quantities of data from all of these in order to decide what's our best candidate for translation into the clinic. Because you can do beautiful things.
1 minute. 1 minute.
Yeah. Okay. I can stop here. I think so. Try multiple assays, see what gives you the best results, and go both into primary treatment choice and into monitoring using the liquid biopsies. Thanks.
Thank you so much. Why don't we bring up the panel? Yeah, there we go. So we have a couple of questions. First, we overlap in chat, and maybe Daria, see if you could take this first, a broad question on, which is, what needs to change to allow these exciting advances to reach the clinical arena? And how could, I'm going to edit here, how could BloodPAC help achieve this?
I think there are, obviously, the first barrier is just complex underlying biology and being able to parse through what that means through the data lens with which we are trying to understand that biology. I don't think anybody can help with that other than thinking about how data could be, data value can be maximized to fully understand that biology across multimodal landscape. And I think what's emerging in the field with more sophisticated modeling capability and hyperparameterized models is ability to integrate these multiple data sources, do inference from that in a way that wasn't easily possible before. So we're kind of finding these things. They're not in the clinical and regulatory part of the landscape, but they are in the landscape of understanding the underlying biology. And I think we need to continue to be very diligent about how we interpret this data in the field. There's it's a lot easier to produce your first positive result than have that result withstand the test of validation and independent data and clinical relevant data sources. So we need to be honest to ourselves about what we produce, because otherwise we're just in the tailspin of chasing things that are not real. So that's what we can do as a community, is being very aware of these two aspects. I think bringing it to clinical practice is another arena where collaborations between treating physicians, academic partners who are at the forefront of understanding that complexity, and you can bring that to patient in a safe manner. Tuvik gave us wonderful examples of how it's been done with some of these cases with patients. But being able to integrate with baby steps in that arena to open up clinical trial opportunities for things that continue to be promising in this setting. And I think we need to balance the level of evidence we need to generate to support these signatures in clinical use with the risk profile that we bring to the patient, and continue to do it in a active exploration mode with the right minds at the table and not go by the sort of trivial checklist sometimes, right? So what is the right level of scientific risk assessment that needs to be in place to keep safety for the patients while being able to bring innovations faster? So to me, that's kind of the facets that are in my mind in order to be more successful as a field.
Thank you, Daria. So I think I'm going to close the Q&A at this point. So I apologize to panelists who prepared to respond to a particular question, but I will note that, as you know, subject to our seminar, we publish all of the Q&A, and so we'll add the additional responses in so you can see those going forward. Shannon, why don't you bring up the blood pack proposed deliverables list? So, as you know, one of the objectives of each New Front seminar is to identify potential areas of focus for future blood working groups. So here is this year's suggested list of proposed pack deliverables. Bridge discovery and clinical implementation. And we would do this by collaborating with regulatory stakeholders to define framework scaffolds that enable translation of multimodal technology innovations into clinical practice. And we would incorporate industry perspectives to ensure pathways reflect real-world development and adoption of challenges, and include consideration for emerging technologies and modal evolutions. Deliverable number 2, advance regulatory dialogue on complex multimodal approaches by providing unified industry input to guide standards for increasingly integrated diagnostic landscapes, multi-omics, AI-driven analytics, etc. Deliverable number 3, expand blood pack multi-omic framework beyond blood pack-based assays by evaluating opportunities to integrate additional modalities such as digital pathology, tissue-based profiling, and other genetic or phenotypic data sources, and developing strategies to merge blood-based liquid biopsy data with complementary modalities for comprehensive biomarker assessment. And the final deliverable, advanced data integration and validation strategies by defining approaches for cross-platform data comparison, method validation, and clinical trial design supporting multimodal technologies, and developing frameworks for integrating genomic, such as liquid biopsy data and digital path data to enable robust analytical and clinical validation. So those are our proposed deliverables, but as always, we ask the audience to, if you have thoughts on what we could do to impact the industry about this topic of integrated multimodal data integration and therapy selection, please provide a suggestion in the chat. So now to close out our seminar, I turn it back to our fearless leader, Lauren.
Hi, everyone. Thank you so much, Jim. Thank you to all of our speakers, all of the panelists. This was a wonderful session. We, as we always promise, to be done exactly at 3 o'clock. If you do have any additional questions for us, please feel free to email Doris or me, and we will actually get them answered and publish them in the white paper that will be available probably at the beginning of January and then posted on our website. So the team will meet again to wrap everything up. We are incredibly grateful to Asaf and to Jim for helping to lead and moderate the conversation today and get everyone organized. And again, a tremendous thank you to Doris for putting everything together and all of her hard work over the course of the past year. Thank you, everyone. Have a wonderful rest of the week, and we look forward to seeing you soon. Take care, guys.